Welcome back to the deep dive. Today we are cracking the secret language of, well, the stationary giants all around us.
We're talking about plants.
Exactly. And unlike us, they can't just pack up and move when things get tough.
And that's really the entire premise of plant survival, isn't it? Immobility, since they can't escape a drought or a neighbor blocking the sun or even a hungry caterpillar.
Right, there's no flight in their fight or flight response.
None at all. So they have to respond by drastically altering their growth, their development. It's all about chemical survival.
And if they can't use movement, they have to use chemistry. So our mission for this deep dive is to unpack the stack of sources you sent over and really get to the heart of these critical chemical signals. We are talking about phytohormones.
Phytohormones, yeah, or plant growth regulators. What's so fascinating is that these signals work in just tiny trace quantities.
Really small amounts.
Incredibly small.
Yeah.
And they control, pretty much every aspect of a plant's life, from being an embryo all the way to senescence, which is, aging.
Yeah. And what's wild is just how different they are from our own hormones. When I think of animal hormones, I picture these really complex structures made in specialized glands.
All right, the endocrine system.
Yeah.
Ferried around by our blood.
Yeah.
Plant hormones are, by comparison, elegantly simple. They're simple organic substances, and they're not just made in one special organ. They're produced by nearly every single cell.
Wow, okay.
And instead of blood, they just diffuse or get transported through the plant's plumbing, the xylem, and the phloem. They're everywhere, basically ready to go the moment a stress signal arrives.
Okay, so let's unpack this chemical toolkit. Where do we start? I think with the first big discovery, right?
Auxin.
Auxin,
Mm.
the master of directed growth. His discovery is this legendary story of scientific detective work. It all starts with observations of tropisms.
Tropisms, which are just curvature responses like bending toward or away from something.
Exactly. And in this case, we're focused on phototropism, bending toward light.
And the story begins with Charles Darwin and his son Francis way back in 1880. It does. They were looking at these grass seedlings, and they had this one crucial insight. The plant only bent toward the light if the very tip of the coleoptile, that little protective sheath, was intact.
So if you cover the tip, nothing happens.
Nothing happens.
Mm.
That one observation proved that the tip senses the light, but the actual bending happens way down below it. So they figured, okay, the tip must be sending some kind of mobile signal downwards.
That's a classic hypothesis.
Yeah.
So then Peter Boysen-Jensen comes along in 1913.
And he's the one who proves the signal is, in fact, chemical. He physically separates the tip from the stem with tiny barriers.
What did he use?
He used gelatin, which chemicals can pass through, and the plant's still bent. But when he used a little sliver of mica, which is impermeable,
No bending.
no bending at all, he proved it was a mobile chemical substance.
But he didn't isolate it. That was Frits went in 1928.
Yes, Frits went. He did this brilliant experiment where he figured out how to extract the chemical by just placing the cut tips on little agar blocks.
So he just let the chemical, whatever it was, diffuse into the agar.
He did. Then he took that block and placed it off-center on a cut shoot that was kept in the dark.
Ah, so no light involved in this part.
No light at all.
Yeah.
And because the chemical was only touching one side of the shoot, that side grew faster, causing the whole shoot to bend.
Even without a light source.
Exactly. He named it Auxin, which means to increase. He proved that it's the asymmetric distribution of auxin that causes asymmetric cell elongation. That is what physically steers the plant toward the light.
That distribution is everything. So how does auxin actually tell a cell to start stretching out so fast?
It's all about molecular architecture, and it's governed by pH. We call it the acid growth hypothesis.
Acid growth, okay.
Auxin stimulates these little proton pumps in the cell's membrane. These pumps start actively pushing hydrogen ions out of the cell into the cell wall.
Which would make the cell wall more acidic.
Drastically. It lowers the pH. And that drop in acidity acts as the trigger.
A trigger for what?
It activates enzymes called expansins. Think of them as, tiny molecular scissors.
Okay, so they're cutting something loose.
They're loosening the rigid fabric of the cell wall. They snip some of the cross links holding it all together.
And with the wall loosened.
The cell's internal water pressure, the turgor, just takes over. The cell can rapidly absorb more water and expand, and that's what lengthens the shoot.
That is an incredible mechanism. A simple shift in pH is the switch that directs a plant's entire movement. And I imagine this had some immediate practical uses.
Oh, absolutely. Massive. The synthetic auxin IBA, indole butyric acid, that's what you buy to dip plant cuttings in to get them to grow roots.
Right, the rooting hormone.
That's the one. And we can also use synthetic auxins to induce parthenocarpy.
Which is seedless fruit formation.
You got it. In crops like tomatoes or cucumbers, you can basically trick the plant into thinking it's been pollinated, and it'll develop a fruit without any seeds.
So on flip side, can you overdose them?
You can. And that's why compounds like 2004-D are such effective herbicides. They cause broadleaf weeds to basically grow themselves to death, but they don't harm grasses like corn or your lawn.
Okay, let's move beyond auxin. You mentioned 6 major classes. The first few are mostly growth promoters. What's next after auxin?
Cytokinins. Their name gives it away. They stimulate cytokinesis.
Cell division.
The very same. They're the hormones that tell the plant to make more cells. You find them in really actively growing tissues like roots, embryos, fruits.
So if auxin is telling the cells how long to get, cytokines are telling them how many to make.
That is a perfect way to put it. And they work together in this beautiful synergy. The ratio between auxin and cytokinins really determines what the cells become.
So like a high auxin to cytokinin ratio might.
Usually forms roots. Flip that ratio, high cytokinins, low auxin, and you'll often stimulate shoot and leaf formation. It's like a 2-part chemical dial.
But the sources also said they have anti-aging properties, which sounds kind of counterintuitive for something that's driving all this growth.
It does, right? But what they do is actively inhibit protein breakdown. They also mobilize nutrients from older parts of the plant toward the younger growing parts.
So they keep the important bits younger for longer.
Exactly. It slows down the whole aging process. You can literally spray cytokinins on cut flowers, and they'll stay fresh for much longer.
That's incredible. Okay, so we have elongation and division covered. What about the gibberellins, or GAs?
GAs are all about vertical growth. They are the hormones of height and waking up seeds.
Hormones of height and scale, I like that.
They stimulate both cell elongation and cell division, so they really drive this rapid expansion. A classic example is bolting.
That's when a plant suddenly shoots up a flower stalk, right?
Well, that's it. That rapid growth is driven by gibberellins, and commercially this is used all the time. They're sprayed on Thompson seedless grapes, for example.
Why? To make them bigger.
Bigger, and it also increases the distance between the grapes on the cluster, just makes for a better, more marketable bunch of fruit.
And their role in waking up seeds, germination.
Right. So a seed first needs water. Once it soaks up enough water, the embryo releases gibberellins. That GA signal tells the surrounding tissue to start breaking down stored starches into usable sugar.
To provide the energy for the new seedling.
Exactly. It's the go signal.
Okay, and the last of the big promoters, brassinosteroids. They seem important, but are often overlooked.
They are, and they're chemically quite interesting, actually. They're similar to animal sex hormones like cholesterol.
Really?
Yeah, and their roles are super broad. They help with cell elongation, cell division. They promote the development of the plant's plumbing, the xylem, and they slow down leaf drop. They're kind of the structural engineers.
Okay, so we've covered the internal chemistry that says grow. Let's shift to how these stationary organisms actually read the outside world. How do they process information like light and time?
Well, light is the master timer. The whole process of light triggering growth and development is called photomorphogenesis.
We've all seen this. If you grow a plant in the dark, it undergoes etiolation.
Right, it gets all pale and spindly and unhealthy, just desperately stretching to find any light source.
And as soon as you give it light, it undergoes deethylation, it greens up and starts growing normally.
And to manage all of this, plants rely on specialized pigments called photoreceptors. They're like the plant's eyes.
And there are 2 main types here, right? Blue light sensors and then the red light ones.
Exactly. The blue light photoreceptors handle the really fast stuff, quick responses, like the phototropism bending we talked about earlier, or the rapid opening of stomata on the leaves.
But the really ingenious ones are the phytochromes. Those are the red light sensors.
They are, and they exist in 2 different states which can flip back and forth. They're photoreversible.
So there's a PR form and a PF form.
Right. Red light, like normal sunlight, converts the inactive form, PR, into the active form, PFR. And PR is the trigger for things like seed germination.
Okay, but then far red light, which is at a slightly different wavelength, flips it back.
It converts the active PFR back to the inactive PR. So what matters is the last light the plant saw. That dictates the outcome.
Which leaves this brilliant strategy you mentioned called shade avoidance. How does that work?
Well, think about a plant growing under another bigger plant. The leaves of the canopy absorb most of the red light for photosynthesis.
But they let the far red light filter through.
Exactly. So the plant on the bottom detects a much higher ratio of far red to red light. Its phytochrome balance shifts heavily toward the inactive PR form.
And that shift is a signal.
It's a signal that says, hey, you're losing the light rays. Grow tall, fast, and get out of this shade. It triggers vertical growth.
So it's not just about how bright the light is, but the actual quality of the light tells the plant about its competition. That's amazing.
It is. And this system is also tied into the plant's internal clock.
Circadian rhythms.
Just like us, plants have these internal clocks that run on roughly a 24-hour cycle. The phytochrome conversion at sunrise and sunset basically acts as the cue that keeps that internal clock synchronized with the actual day.
And that perfect timing is what allows for photoperiodism, right? Knowing when to flower based on the time of year.
Yeah, and for a long time, people thought it was about the length of the day. But research in the 40s showed it's actually the length of the uninterrupted night.
The critical night length.
Right. Short day plants need a long, uninterrupted night to flower. Long day plants need a night that's shorter than a certain maximum. The phytochrome system is what's measuring that period of darkness.
Okay, we've covered growth and light. Now for the high-stakes stuff. Survival chemistry. The inhibitors that tell a plant to shut down and defend itself.
And that brings us to abscisic acid, or ABA. It's often called the stress commander.
The emergency brake.
That's a great way to put it. It's the primary signal for managing bad conditions, especially drought. When a plant senses water stress, ABA levels just skyrocket.
And what does that surge of ABA do?
The first and most immediate thing is it causes the stomata on the leaves to slam shut. This drastically minimizes water loss through transpiration. Without this, a plant would just dry out and die in hours.
But it's also crucial for seeds, isn't it?
Oh yeah, it's the gatekeeper. ABA is responsible for seed dormancy. It's a chemical safety lock that ensures a seed only germinates when conditions are actually good for survival.
And it also triggers deeper molecular rescue missions inside the cells, right?
This is where ABA really shines as a crisis manager. High levels of ABA turn on all these defense genes. First, it activates antioxidant enzymes, things like SOD and CAT, to clean up toxic byproducts of stress.
It's like an anti-inflammatory for the cell.
A potent one. Second, it promotes what's called osmotic adjustment. It tells the cell to accumulate things like proline and sugars.
And those solutes, they help the cell hold on to water.
Exactly. They lower the internal water potential, allowing the cell to maintain turgor and keep functioning even when there's not much water around.
Now, the effect on roots is more complicated, isn't it? It's not just a blanket stop growing command.
It's biphasic. It's really strategic. Under mild stress, low concentrations of ABA actually stimulate root growth, telling the roots to go search for water deeper down.
A third party.
Yes. But once the stress gets really severe and ABA levels are very high, then it inhibits root growth to conserve resources.
And that's not just ABA acting alone. This is where that hormone crosstalk comes in.
It is. The high ABA levels inhibit root growth, partly by promoting the synthesis of ethylene and by messing with auxin transport. The three of them work together. No hormone is an island.
Speaking of, let's talk about ethylene, the gaseous hormone, the one associated with endings and stress.
Ethylene, the simplest alkene gas. It's produced really fast in response to all kinds of stress injury, flooding, even just mechanical pressure.
Like a growing shoot hitting a rock underground. That triggers the famous triple response.
It does. And it's like the plant hits the brakes, inflates its tires, and turns the wheel all at once. The shoot slows its elongation, the stem thickens up to get stronger, and it starts growing horizontally to get around the obstacle.
That's a great analogy. Ethylene is also, of course, the hormone of senescence.
It is. The hormone of programmed endings. A burst of ethylene is what controls leaf drop in the autumn. It's actually this delicate dance between ethylene promoting the drop and auxin trying to inhibit it. When ethylene wins, the leaf falls.
And for anyone who buys fruit, ethylene is the reason it ripens.
Yes, and this is where you get a powerful positive feedback loop. Ethylene triggers ripening, and the ripening process, in turn, releases more ethylene.
So one rotten apple
Can absolutely spoil the whole barrel. But it's also how growers can pick fruit green and then control the ripening perfectly with ethylene gas during transport.
Okay, last thing on defense. Plants can literally call for help.
It's one of the most sophisticated defenses. When a plant is being eaten by, say, a caterpillar, it can release these volatile organic compounds into the air.
An airborne distress call.
Essentially, yeah. And that signal can attract predatory animals, like parasitoid wasps, that will then come and attack the caterpillar. The plant basically calls in air support.
It's just amazing. So let's synthesize this. What's the core takeaway from this whole deep dive for you, for the listener?
I mean, for me, it's that the entire complex life of a plant is governed by just a handful of simple organic molecules in tiny amounts. And understanding these pathways isn't just academic, it's critical for sustainable agriculture.
You mentioned the ABA receptors specifically. Understanding the nuts and bolts of that must be huge for developing more resilient crops.
It's everything. Researchers are intensely focused on these receptors. They go by PY, RPYL, CAR. Think of them as the lock on the cell, and ABA is the key that shuts the stomata.
So if we can engineer plants with a more sensitive lock,
Or plants that make the key faster under stress, we can dramatically improve drought tolerance. And with a changing climate, that is essential.
it really is. But thinking about ABA's role in extreme stress, it seems like it forces an almost philosophical choice on the plant.
It really does. Because ABA doesn't just help a plant endure a drought. Research shows that if the drought is early and severe, these high sustained levels of ABA can actually activate the main flowering genes.
Wait, so the stress hormone makes the plant flower?
Yes, it forces the plant to rush through its entire life cycle to produce seeds as quickly as possible. The plant basically makes a decision to initiate drought escape.
So it's giving up on the endurance battle.
It's deciding to finish the race. It's a strategy to ensure its genes are passed on before the conditions become completely unsurvivable.
So a plant has two choices: hunker down and try to wait out the drought, or hit the emergency button and complete its life cycle before it's too late.
Which leaves you with a pretty big question, doesn't it? For an organism that can't run away, is survival always about endurance? Or is it sometimes about knowing exactly when to finish the race before that final, inescapable disaster hits?
A profound chemical ultimatum, definitely something for you to mull over on your own deep dive. Thanks for joining us.